Hello, everybody. We're going to do a really quick review of DNA. So the difference between DNA and RNA, you can either look at my scribbled diagram here or the pretty one that I found on the internet. But either way, the difference is that DNA and RNA are both made of nucleotides, but DNA is double-stranded and it stays in the nucleus. It has four bases, A, T, C, and G, and the sugar that DNA is made of is deoxyribose. RNA is a close chemical cousin of DNA. It is single-stranded. It is made of ribose sugar, therefore R for ribose sugar, D for deoxyribose sugar, and the four bases are almost identical. The only difference is that thiamine is replaced by uracil in RNA. So let's talk about the processes of DNA. First one that we're going to talk about is DNA replication. We're making more copies of DNA, and that's occurring in the nucleus. This is semi-conservative, meaning that every time we copy DNA, we're making one old strand connecting to one new strand, instead of having one double helix that's all old stuff and one double helix that's all new stuff. They're a mixture, one old, one new strand twisted together to make a double helix. So the first phases of DNA replication involve taking that twisted up double helix and unzipping it using helicase. Helicase works on the helix and it unzips your genes. Then along comes DNA polymerase, and polymerase is our builder, and it's going to build the matching nucleotides, the C's, G's, T's, and A's of DNA, and it's going to match them up to the single naked strand of DNA here, and it's going to put together a double helix. On one strand, the leading strand, everything happens super smooth and it occurs moving toward that replication fork where DNA is split in half. The other strand is known as the lagging strand, and it moves away from the replication fork in short chunks. It'd be so convenient if DNA worked in the same direction, but they're opposite strands. They work in opposite directions. So when you do that, you're going to have these gaps. And ligase is our third enzyme, and its job is to come along and fill all of those gaps on the lagging strand of DNA. So when you're doing that, the complementary nucleotides in DNA are A and T will always be across from each other, TA, CG, and GC. So A and T are always partners, C and G are always partners, and a good way to remember that is that C and G, the letters look alike, so they're going to be close to each other. The next step is to make RNA. If we think of DNA as a giant cookbook, RNA is the single recipe card that we need to make one thing, one type of cookie, or one type of barbecue sauce, or whatever that you have a recipe for. So we have one enzyme here involved, and its name is RNA polymerase. And polymerase is going to unzip and build as it unzips. So we don't have to unwind the entire double helix, we just have to unwind it for a short segment as we read and copy, and then we let it seal back up again. We're not completely splitting it in half. When you're doing this, you're copying the A's, T's, C's, and G's of DNA into a simpler single-stranded message of mRNA. And the A and T want to partner up, but RNA, as we went through on the very first slide, doesn't have the letter T. It has the letter U to replace it. So across from every DNA letter A is the RNA letter U. So here we go. CCA, the G's partner up, but A partners up with U instead of T. And then everything else looks pretty much exactly the same. So if you had a strand of DNA that read ATCG, the matching RNA that would be built from that is UAGC. Oops, wrong button. We can make three types or shapes of RNA. mRNA stands for messenger RNA. It is a very simple copy of the DNA message, and we're going to read it in three-letter chunks called codons, and we think about mRNA like the recipe card. These are the instructions for how to assemble a protein. The second type is tRNA, or transfer RNA, and its job is to bring the correct ingredients. So it's going to bring these amino acids, and it's going to make sure that they match using an anticodon. So three letters at the bottom of this tangled up structure match three letters here, and that's how we double check that we've brought the correct thing. And then the third type is the rRNA, or ribosomal RNA, and its job is just to build up and bulk up a ribosome. The last step is to take that RNA and actually assemble a protein at the ribosomes. And ribosomes can be found either in the ER or in the cytoplasm of a cell. So to do that, we take this mRNA message, this code, and we pull it through a ribosome like a tape dispenser. And we look at it three letters at a time, or a codon at a time, and that tRNA will drop off the matching amino acid if the codon and anticodon are a perfect fit. So as you can see, across from every C is a letter G. That's a perfect fit. So we're going to drop off that amino acid. We're going to link them all together, and we're going to assemble the ingredients of the protein in order. When you are doing this process, what you're going to look at are those mRNA codon letters and this chart. You will have access to this chart. You don't need to memorize it or anything. You can use it on the test, but you're going to start in the center, like a bullseye, and work your way out until you find the correct amino acid ingredient. So if the mRNA code reads AUG, we start here with A, then U, then G, and we know that the first amino acid we're going to grab is MET, or methionine, the start codon. Then CCC. C, C, C leads to proline, and proline is going to be our second amino acid ingredient. GUG is our third one, so G, U, G leads to valine, and that's our third amino acid ingredient. And you would just keep going using that exact same pattern. You don't have to do anything with it. If you see the mRNA already, you just feed it directly into this cycle. If you started with DNA, you'd have to do a couple extra steps. The last thing that we wanted to talk about is mutations. There's a bunch of different causes for mutations, but we break them down into a couple simple types. You can either have a point mutation or substitution, where one of the letters is just swapped out for a different one. You could also have a deletion, which is where one of the letters is eliminated and cut out of the DNA strand, or you could have an insertion, where extra things are added in. If you have a point mutation, chances are you're just changing one amino acid because you've only altered one codon. Instead of CTG, now you have CCG. So you're going to make a slightly different piece using these letters. Now, if you have, if you have a deletion or an insertion, as you can see here, all of the rest of the DNA has to shift over, which means that every codon or sequence of 3 letters is going to be affected, which means that we're going to be bringing the wrong amino acids and assembling the wrong ingredients for the protein, which can cause a lot more problems, which is why frame shift is so bad. That's a really quick review. I hope it helped you guys, and that is all.